All right, welcome back. We are going to do some real basic genetic um, teaching. All right, just give you the kind of the basics of this. So this is Mendel. So Mendel is the father of genetics. He was a monk also. He worked in the ministry where he could do his research as well while he practiced his faith. He worked on many different organisms. He's famous for his peas, but he didn't just do peas. Peas just worked really well. Like I said, experimented with animals and different types of organisms, but peas worked the best, and he stuck with that once he discovered it. So the thing that works really well with plants is that, in particular, these pea plants, they're what they call a perfect flower. A perfect flower has both male and female parts to it. A lot of flowers have both. So we can see we have the male anther and the female stigma, the male and female parts. And so the pollen is on the little tip there. It will then land on the top of the stigma, and then it will grow a pollen tube down and then fertilize the eggs. Once the eggs are fertilized, those are seeds now. They will, if they're planted, grow, and now we have the next generation. So for Mendel, all he had to do was, I say, neuter his flowers. He snipped off the male parts. He could then, he's in a greenhouse, he could control by snipping off the male parts. He could put them in jars, and then he could control which plants crossed with which plants. He could just take a little paintbrush, he could get the pollen off and put it on the female parts, and then observe the baby peas. Now, there are two types of pollination in plants. There is self-pollination, so like this flower could pollinate itself, or the pollen can go to another pea plant and pollinate a different pea plant that may not be identical to itself. That's called cross-pollination. So self-pollination versus cross-pollination. These are some of the traits he looked at with peas. He looked at whether they were smooth, wrinkled, yellow, green for the actual pea versus the pod color. We also have the pod texture and the pea texture. So we have the smooth, wrinkled, the flower colors. Where are the flowers positioned on the plant? And was the plant tall or short? So when we look at things like, okay, is it purple or is it white? That gene is for flower color. Now, the gene has, in this particular case, two options for flower color, purple or white. That means there's two forms of the gene that will give us two different colors of flowers. Forms of genes are called alleles. So we have an allele for purple and we have an allele for white. We have an allele for tall, we have an allele for short. Some of the other vocabulary is genotype versus phenotype. So the genotype is the actual gene itself. Now, we're not going to give you a DNA sequence. We're going to give you a letter to represent that gene. So in this case, we're going to use T for tall, big T for tall, little t for short. The phenotype is what we actually see. Now, when I look at a particular plant, I'm not going to see that, oh, that plant is big T, little t. But I am going to see that that plant is tall or short. So the observed characteristic is the phenotype. The instructions that give you that characteristic is the genotype. So for example here, every gene has two copies, one from mom, one from dad. So if you get the gene for yellow pea colors, let's say we get the same yellow for mom and dad, we're going to call that individual homozygous dominant. Homo means same. So we're saying they have two of the same dominant genes. Over here, we're going to use homozygous recessive. So they got that recessive gene from both parents. Right? So which is the only way I can see the recessive phenotype is if they're homozygous recessive. If we are hetero means different. If you're heterozygous, that means you have one, both of your copies are different. So this individual has a yellow copy and a green copy. Now, the P is yellow because of dominance versus recessive, which we have right here. If there is a dominant gene, in this case, yellow is the dominant, it will hide or mask the recessive. So if we go back to this picture here, let's see if I can get all the coloring done. Okay, I can't. So if I can get, to get these colors and stuff on which is dominant and which is recessive, I'm surprised there's not an eraser. Okay, you have to actually breed. You have to actually breed those particular. Ooh, there we go. I found it. I'm so excited. You have to breed those pea plants. He's not just going to say, oh, I want purple to be dominant and white to be recessive. No, he had to breed the purple, homozygous purple, with a homozygous white to get the hybrid, heterozygous, and then see what color it turns out. Whatever color is shown, that is the dominant. One that's not seen is recessive. It's being hidden. Okay. So we have 3 different types of genotypes: homozygous dominant, homozygous recessive, and then we have our heterozygous. Okay, hetero means one dominant, one recessive. Now the phenotype, now notice we have these, all 3 give us yellow, right? Because it's going to cover up the recessive. The only way I can see a recessive trait is to be homozygous recessive. So when we set up these crosses, we're going to have what are called the parentals. The parentals are generally purebred. All right, so like here when I say green, so I would have, let's do this. That would be little g, and yellow would be big G, big G. Okay, they're purebred. When we cross those, the F1 are going to be yellow because they're going to get one from each parent, right? So we're going to get one of here, one here. And so the offspring, the F1s, are going to be hybrids or heterozygous, and they're going to show that dominant trait. Now, Mendel self-pollinated his flowers over and over and over to get purebreds. And then he started seeing what the F1s, and then the next generation, the F2s, were like. So we have this lovely tool, I'm sure you learned this in high school, called a Punnett square. It's called the Punnett square because Mendel's work sat for many, many years and people just couldn't understand what he was trying to say. He was a genius. People could not interpret what his work was saying. It wasn't until Reginald Punnett, he was a statistician, picked up his work that he could read it and understand it and then put it in a way that we could understand it. And that's why it's called a Punnett square. So if we have our here our homozygous dominant, here we're looking at pod color, not P color, versus our homozygous recessive. So those are our purebred, and we cross them. So you're going to take the little g down and the little g down, and then you're going to take the big G across and the big G across. And you can see all the F1s. This is going to be your F1 generation. They have the same genotype, big G, little g. They're all heterozygous. And they're all going to look the same. They're all going to be looking dominant. They're going to have that dominant color. Now, if we were to take, let's just say our F2, we were to take that heterozygous one here and cross it with a homozygous recessive, okay, we can do our cross again. So we have our big G going across, and then you can see the colors, you know, the yellow, our little G goes across. And then we can take our little G down and our little G down. And we can see here where we're going to get half are going to be green. and half are going to be yellow. Okay. So this is why sometimes when you have these recessive traits hiding here, you may not see it for a while until the other parent may either have that recessive characteristic or they may be a carrier. We call them carriers also. And then it gets expressed. We see it. Okay. And we talked about those different combinations during meiosis, right? We can have, whether it's the law independent assortment or crossing over, that we get these different types of variations. So here we have our parentals, which were purebreds, and then we have our F1s, right? Because remember during meiosis, we're going to go from 2 copies to 1 copy. And that's basically what we're saying here. Well, that's what we're saying in this Punnett square. If this Punnett square was big Y, little y, we have to separate our alleles, right? So big Y is going to go here and the little y is going to go here. So 50% of that individual's gametes will have a big Y and 50% will have a little y. That's what the Punnett square is saying. Okay. This is where we're segregating and we're separating, we're going through our divorce. And then we come back together during fertilization and we get an F1 that is heterozygous and yellow. And then again, we're going to separate those out again during meiosis to make our gametes. And then if we do a self-cross, so if we cross this with this, okay, notice we get our green back. All right, so we went from being all yellow, we didn't see any green in the F1s till we'll get about 25% are going to be green in the second generation, the F2s. Okay. So that is my genetics in a nutshell. You will have some reading in your textbook and some videos to help you along with this. And hopefully this helps you understand some basic genetic ideas. If you have any questions for me, please reach out and I will see you in the next video.